Vanakkam. Welcome to this course on Classics in Neuroscience. In the last weeks, we looked at neural circuits, those that mediate different types of perception and cortical processing and cortical columns. In this week, we started looking at neural systems, specifically the neural basis of behavior. In the last video, we looked at the reticular activation system. In this video, we will look at sleep and rapid eye movements, specifically the seminal contributions of Eugene Aserinsky towards improving our understanding of the neurophysiology of sleep. For the longest time, it was believed that sleep is this passive phase where rejuvenation or restoration of the brain happens. Shakespeare, in one of his works, says the same. This was believed for the longest time by everyone, also including the scientists. One of the scientists working in the field of sleep physiology was Nathaniel Kleitman. He supported the traditional view of sleep, which is sleep is a passive restorative state of the brain. The modern study of sleep that suggests that sleep is indeed alternating states of activation that happens in different regions in the brain. This was proposed by one of Nathaniel Kleitman's students, Eugene Aserinsky. Eugene Aserinsky was born in 1921 in Brooklyn, in New York. Essentially from a modest family, and he graduated from high school at the age of 16. He did start college, but somehow I think he lost way and did not really finish college. He did a whole bunch of odd jobs for 12 years. And in 1949, due to the GI Bill, he decided that he will pursue a career in science. That is when he was 28 years old, without a college degree. Formal degree he did not have. But he somehow convinced the University of Chicago to allow him to take the entrance exam, the graduate entrance exam, in which he performed brilliantly. So the University of Chicago allowed to join the graduate program, where he wanted to pursue organ physiology, and he started studying sleep with the mentoring of Nathaniel Kleitman. Let us take a few seconds here and consider, would this be possible today? Without an undergraduate degree, could you get admitted to a graduate degree? Practically impossible, because your transcripts and your graduate record examination scores are all shortlisted by a computer. So if you don't have transcripts, you will be summarily rejected without consideration. So this is what would happen today. So we have relatively rigid systems today when compared with more flexible systems of the past, that actually led to the encouragement of good scientists like Eugene Aserinsky. Something to keep in mind. So Kleitman observed that while an infant falls asleep, this phenomenon was associated with blinking of the eyelids. So Kleitman asked Aserinsky to follow up and find out how and when infants fall asleep. Essentially, he was given an empty laboratory with a reclining chair for the participant to sleep, not for the graduate student to sleep. Okay. Because this is sleep research, the participant will come and lie down and sleep. It is not for the graduate student or the researcher or Eugene Aserinsky to sleep. So there was an empty laboratory with a reclining chair and ancient smoke drum apparatus or a kymograph. And practically Aserinsky worked alone. There was absolutely no collaboration. There was no concept of having colleagues within the lab or lab mates. That concept was not there. That is the situation practically in many different parts of the world, in many different labs. So, that was pretty much common, or at least not that uncommon in those days. So, Aserinsky observed these blinking movements in infants, and he found there was practically no pattern from these blinking movements. Since there was no pattern in blinking movements, he decided to observe all movements throughout the night. Just imagine this, you have to observe all movements of infants throughout the night and analyze this data during the day. It was a very tough phase of his life, because by that time he was already married and he had 2 kids. His wife was suffering depression, and he himself was not making that much money to feed the family. Frequently the family was left without food. This is how hard it was. But he found some interesting results at that time. He found that there are indications that suggested there were some specific 20-minute no eye movement periods. There were some periods during which there was absolutely no movements in the eyes. He then decided to pursue similar experiments in adults. By that time there was also some new equipment that arrived in the lab, some old electronic polygraph with multiple channels of electrodes, what is now called as a electrooculogram, and also EEG. This polygraph was not that good, it was somewhat unreliable, and careful scientists, they are always worried about artifacts. After removing all the artifacts, he found that eyes undergo specific saccade-like movements, very fast movements, saccade-like movements. The eyes are closed. Why would the eyes move in closed eye? That is the question. So, whenever he observed these kind of fast or saccade-like movements in his participants, he began waking up his participants, asked them if they were dreaming, as if their eyes were moving, as if they are quickly watching something, watching a moving object or a moving animal or something, that is watching a movie, for example. So, finally he confirmed that these 20-minute rapid eye movement periods are associated with a dreamy state. Then there was an upgrade in the equipment, so he also could record the movements with a movie camera, and there was also another medical student who came to assist him, William Dement. So, finally after his observation, the pattern that came together was there was a falling asleep period, then there was a, after some time there was a rapid eye movement period, then there was a no eye movement period, then there was another rapid eye movement period, then there was a no eye movement period. So, there were alternating rapid eye movement period and no eye movement period that happened one after the other. He reported his findings in the annual meeting of the biologists in Atlantic City and published in Science a paper titled Regularly Occurring Periods of Eye Motility and Concomitant Phenomenon During Sleep. So, in that paper he described rapid, jerky, binocularly symmetrical eye movements, the increased ability to recall dreams that occurred in the presence of these movements, increased rate of respiration and heart rate too during these periods. It was as if people were watching an intense movie. So, they could recall the dream, the heart rate was up, the respiration rate was up. The full paper came later in 1955, I think, that was titled Two Types of Ocular Motility Occurring in Sleep in the Journal of Applied Physiology. This acronym REM, rapid eye movements, gained a lot of traction. Even today, REM, if you say, people understand what that means. So, this was first reported by Eugene Aserinsky. So, there was a need for Aserinsky to move on from this life, and he, I think, took a break from sleep research, and I think he joined Jefferson College as a faculty member. Dement and Clayton continued this line of work, and later, maybe almost a decade later, Aserinsky returned to this area with another publication where he described the physiological differences between REM sleep and non-REM sleep. So, sleep initially, as described by Shakespeare, being the passive resting, restorative, rejuvenating state of the brain, later, after Eugene Aserinsky's experiment, became alternation of actively interacting brain regions and circuits. Later in the 1970s, Michael Jewett characterized different brain regions and their neurotransmitters involved during sleep. So, it is not like the entire brain is sleeping during sleep, that happens in coma or death. And Alan Hobson and John McCarley identified activity in different brain circuits or alternating brain circuit activity supporting the different stages of sleep. So, there are stages of sleep during which different brain regions get activated. With this, we come to the end of this video. In this video, we looked at how sleep went from previously being seen as this passive, rejuvenating, restorative, restful phase to a phase in which there were alternating activation and deactivation of specific neural circuitry within the brain, leading to observable differences in behavior. For example, in terms of the rapid eye movements and no rapid eye movements. Essentially, the contributions of Eugene Aserinsky to improve our understanding of the physiology of sleep. Thank you for your attention.